Azolla. Azolla is a water fern that has been traditionally used in the biofertilizer in the rice fields of South and Southeast Asia. And this is due to the property of nitrogen fixation of a cyanobacteria Anabaena azole that is present inside the plant body of this water fern Azolla. And in fact, due to this reason, the symbiotic relationship, the Anabaena and Azolla, they are in a symbiotic relation where the Anabaena provides fixes nitrogen and the Azolla photosynthesize, and each of these symbiont use the products produced by the other. And in this process, when the Azolla decomposes the fixed nitrogen, it is used up by the rice plants. Hello friends, I am Dr. Ayantika Das, and in this video I am going to discuss about another pteridophyte, another aquatic pteridophyte, Azolla species. And in this video will be in two parts, and I'll be discussing the overview, the distribution, morphology, anatomy, ecological and economic importance, and the fossil forms of Azolla in this part of the video, that is the part 1. And in the next part I will be discussing the reproduction and life cycle. So let's begin the video. So taxonomically, Azolla has almost the same position as that of Salvinia, and it belongs to division Polypodiophyta, then class Polypodiopsida, that is common to all ferns. It belongs to order Salviniales and family Salviniaceae, and another aquatic fern Salvinia also shares the same order and family. And our topic of discussion, Azolla, is a water fern, but there are certain difference between Salvinia and Azolla, especially in their external morphology as well as the internal, that is the anatomy portion. Both are water fern, both are floating, but there is a basic difference in both morphology and anatomy as well as the reproductive bodies that are born in these water ferns. Azolla has it's very invasive by habit. It's very invasive. In fact, this is a plant that has been introduced. It looks so pretty on the water surface, growing on water surface. It has been used as a ornamental plant in gardens. But if they due to their property of excessive growth, they have spread through as a invasive weed in most part of the world where it has been introduced. So by habit it is very invasive and high growth rate, and that is why it can alter the biodiversity of the aquatic system. This is mainly because of the fact that it can grow so fast that it covers the entire surface of the water body. So the for photosynthesis, the sunlight is cut off, and those submerged plants, they cannot photosynthesize in case of an Azolla invasion, and they will ultimately die. Thus, any pond that has Azolla in it will not have any or very little benthic or submerged vegetation. So, by habit, it is a freshwater fern, and it is found in area where the water current is very low, because high water current will cause the breakage of the rhizomes or the stem or even the frond. And it can grow over a wide range of pH. It can grow over almost normal tropical temperature, but it cannot grow, it cannot tolerate very cold temperature. That is why in the temperate region it survives in perinating bud condition. And as because azolla is a nitrogen, it contains anabina, which is a nitrogen fixer, it can grow in very low amount of nutrient soil, nutrient water also. Water that is very low in nutrient, it's called oligotrophic water, and this oligotrophic water also can sustain the azolla due to the inherent property of nitrogen fixation that is done by the Anabina azolla. In fact, this Anabina azolla association has been even found in the fossil record of azolla fossils. Some of the species are also salinity tolerant, and they one thing that they are not tolerant is that desiccation. It cannot tolerate desiccation and requires to be grown in water. The distribution, the relative generally of the area should be very close to 90%, not found in the polar region, and we see that it is found in the Azolla filiculoides. It is found in Tasmania. In here it is found, that is in the Australasian Sea, and pinnata, Azolla pinnata, we find it in Asia, mainly South and Southeast Asia, including these small islands also, and Australia, and Africa, and Azolla nilotica. It is found in the Nile region, and that is in the northern part of Africa. Sorry, this is the, I'm not drawing it properly, but we know where Egypt is. So Nilotica is mainly found in the Nile region. Rubra is again found in Australia and New Zealand area. So we have this Azolla in the southern hemisphere also, and Azolla cristata is present in the North America, the central or the Mesoamerica portion, and also the southern America. So we see that it has got a, from temperate to tropical, this region tropical, to the temperate region, it is easily found. So Azolla has got all this, it has got a wide range of, in morphology, these all are the Azolla has got a, where we can it is floating Azolla. And it has got a horizontal stem body, that is the horizontal stem, that is called the rhizome. This is the picture where the rhizome, it is hidden from the view, as because the rhizome has got a covering of 2 rows of leaves that are overlapping each other. Here we see that each of the leaves, they are overlapping, the leaves how they are overlapping, and these leaves, they cushion or they blanket the rhizome, and they are arranged in 2 rows and they are overlapping, and these leaves are present on the upper surface. If this is the rhizome, the upper surface of the rhizome has got leaves in 2 rows, and the lower surface will have roots, and the root, that is, will be we know that most of the ferns, they have a advantageous type of root. The same holds true for Azolla also. And the rhizome of Azolla, which is hidden from view, it is covered by small hair, and the branching is pinnately branched. So we see that the branching is pinnately branched, which means that it will be like this. And one thing about the branching pattern is that the older branches, this is the older branch, this is the oldest branch rather. So the older branches, they are much larger length than the youngest branch. So we see that the branch at the basal region or the oldest is much longer than the younger branch. So as a whole, it gives a triangular appearance to the branch or the body. Here we see the triangular appearance. We see this one, this one. So this type of branching in the all the branching is pinnately branched, just like the leaves of the leguminous plant. In fact, the leaf of the leguminous plants, they are also called fern leaf because of the similarity. And about the branching, then about the breakage. The most of the time, this azolla, their rhizome is so weak that sometimes the rhizome or the branches, they break away, and that fragment give rise to new plant. And in fact, this fragmentation of the branches or the rhizome or any front portion is a very common method for propagation of azolla. That is why it is one of the reason, that is why it is considered very invasive. Even if you harvest the the azolla from your pond, and a small fragment is left, and also that is, it is very minuscule in size, so any small fragment can be left, and we are not able to totally eradicate azolla, because that small fragment will produce the whole bloom the root again of the azolla, as I not told, bloom, that the whole they have an advantageous of azolla root. The primary root that is born, they are ephemeral, that is not long lasting. After a certain period, they are replaced by this advantageous root, and the roots, they hang in water either in cluster, but they are usually even if they are clustered or single, they are usually unbranched. And about the fronds, that is the leaf are bright green in color, but overexposure to sun can cause reddish color in the plants. And this coloration also vary from species to species. And this fronds at very immature stage, it is not circinately coiled. Like in some other type of fern, there is a circinate type of coiling, but such type of coiling, this type of coiling is not present. It is not present. So the leaves, they are not circinately coiled, but the leaves are of equal, there is two lobes. That is the first lobe, which is like round to oval, and equally sized second lobe. And this first upper or the dorsal lobe, this is very important because they contain the mucilaginous cavity that contains the Anabaena, the cyanobacteria. And the lower lobe or the ventral lobe, here it is also very important because the lower lobe will contain the sporangia, or the structure that contains the spores, or in which the spores are produced. So we see that this leaves, they are so closely packed that they have certain amount of air in between them, and this amount of air helps in buoyancy. Why I am saying this? It is because that unlike Salvinia is also another Salviniaceae family water fern. So that Salviniaceae has got aerenchyma tissue in its stem, in its root, but here the stem does not contain aerenchyma. So in order to maintain the buoyancy, here is trapped in between the leaves. The leaves are so closely packed, and in between the leaves, small amount of air space is left, and that air space helps in the buoyancy. The leaf, we see that these are each of the leaf structure, and So each of this leaf, I told you it has got an upper and lower lobe. So the upper dorsal or the floating lobe, it can be of various size and shape, generally ovate and sterile. The size will depend upon the species. And this upper lobe, it is very thick and it is greenish in color. In fact, it is photosynthesizing lobe. This upper lobe photosynthesizes, and that is why it is green in color. And inside this lobe, the upper lobe will contain the cyanobacteria anabena inside a mucilage filled cavity. And only one species, that is Anabena azolla, is present in all the species of Azolla. And the lower lobe, about the lower lobe or the ventral lobe, this lower lobe, it is thin and very transparent colored, and it is submerged. Just because it is submerged, that is, it is under water, so it has got certain absorptive function. So this lobe, the lobe, I'm drawing it in yellow color, then you will understand it better. So this lobe, it will contain sporangia, or the organ that produces spore. So the upper, we see that the upper lobe has a function of photosynthesis and nitrogen fixation, and the lower lobe, it contains the reproductive organ. So we see that both lobe has got different function, and there is compartmentalization of or division of work between the two lobes. Upper lobe is for metabolic function and the lower lobe is for reproductive function. And both the lobes, they are also physically, morphologically also very different. One thing that is, the upper lobe, on the surface, they have got trichomes or hairs, and these trichomes can be either unicellular or bicellular depending upon the species. And this, why I am discussing these trichomes, because trichomes are very important. These trichomes on the surface, you see here also, we see that these small dotted things, these are the trichomes, and in fact these trichomes of Azolla make it water resistant. They do and just because on the upper surface water cannot accumulate, so it helps in keeping the plant buoyant and not getting submerged. So this is the very important function of unicellular or bicellular trichome, which are water resistant. This is the how an Anabena looks. This is the picture. This is the diagram of Anabena. It is a cyanobacteria which is filamentous, you see, and this big shaped cell, it is the heterocyst. It is the heterocyst here, and in fact heterocysts are only this big cell, that is the heterocyst, is only for nitrogen fixation. So this Anabena is present on the upper lobe or the dorsal lobe. And another thing, the ventral lobe contain the sporangia. Now sporangia, that is the organ that produces spores in Azolla, is of two types. So Azolla is a heterosporous fern, that is, it will produce a male, that is the macrosporangia, and a female sporangia, that is the megasporangia. Here are the diagram how the mega and microsporangia look. Morphology or transverse section. On transverse section, we see that the stem has got an outer layer of epidermis. Now after this epidermis, there is few layers of hypodermis. dermis. The epidermis is parenchymatous. The hypodermis, it is different from Salvinia by having no ear cavity. The hypodermis or the cortex, it does not have any ear cavities. Then we come to the stellar portion, where we see that there is a single endodermis. This is the endodermis, and next to the endodermis, inner to the endodermis, is this layer of cell. This layer of cell, it is called the pericycle, and inside the pericycle is the stellar region, where we see that these thick walled cells, they are the xylem, and we see that there are two xylem bundles here, and on the outer surface of the xylem bundles are the phloem bundles. I am drawing it in red. So this is the phloem. This one here is the phloem, and this thick celled xylem. So phloem is on the outer surface, xylem inner surface, and the stele, it is generally protostelic to siphonostellic, and then so about this is about the stem morphology. If we see the transverse section of this is the dorsal leaf. We see that these are the anabina. The filamentous blue-green algae that is responsible for nitrogen fixation. So we see that the leaf transverse section, it has got an epidermis. On the upper surface, it has got epidermis. So on the upper surface is the epidermis. On the lower surface also we have got a epidermis, and in fact the upper and the lower surface both contain epidermis, contains stomata. The upper surface, in addition to stomata, will also contain hairs or trichomes that render it water resistant. And next layer is the mesophyll. We see that there is these are the mesophyll layer with ear space in between. This is the ear space we see. This is the ear space, and these are the mesophyll cells. So we have an erenchyma in the leaf, and inside next to the mesenchyme is the mucilage filled cavity that contain anabina. So this anabina, it is present in the upper that is the upper lobe. And in the lower lobe, where the reproductive organ, the spores are formed. Later the male gametophyte and the female gametophyte all is present in this lower lobe only, and when the new sporophyte is formed from the gametophyte here only, then this anabina from the dorsal region, it passes on to the lower lobe and gets incorporated in the root morphology, embryo, where we see that they have got in this way, from generation to generation, epidermis, anabina can have then the symbiotic relationship. After the epidermis Azolla is the hypodermis, and after that we have the endodermis and pericycle. So this one is the endodermis. Next to it is the pericycle, and then we have our stele. Now stele, here we see that consists of both protoxylem on the outer surface and metaxylem on the inner surface, and we see that the number of metaxylem, it is 2 in number, and the protoxylem is 4 in number, and they have got now phloem elements. This is the most important surface part. Azolla has been put to several use. Azolla has been used as a food and fodder, as also incorporated in various human food also. But one thing of concern is that Azolla it has got certain neurotoxin property. This neurotoxin BMAA, it is found in the body of animals that is fed on a Azolla diet, and after consuming this BMAA, it has got a property, that is, it can pass along the food chain and it can biomagnify. So the problem of biomagnification of BMAA is now rendering Azolla a very doubtful candidate for use as a food, as a fodder, feedstock, or even human food, because BMAA is quite neurodegenerative in their property. Now, about the ecological importance, we see that on because of its much beauty in its external structure, it has been used as an ornamental plant in Europe, and once it has been introduced from Asia to Europe, then it was declared invasive, because it is very high. It has got a very high growth rate. Not only that, it is also damaging the entire ecosystem by reducing the floral diversity in the water body, by shading the all the other plants, and as a result of loss of submerged plant, which are the food for fishes and crustaceans or marine invertebrate or aquatic invertebrate, sorry, not marine invertebrate, aquatic invertebrate. So any Azolla in any ecosystem will totally damage all other, endanger other biodiversity. So that is why it is also ecological concern. And that apart from ecological concern, it also causes certain economic loss also. That is, if you are pumping water from a water body, and that clogs the pump or the filter, so it causes economic loss. Also, they can impede the flow of water in the channel. Also, when excess amount of Azolla, they decompose, they reduce the dissolved oxygen of the water body, so that is also detrimental for the health of the water body. Another ecological importance is its mosquito control, as because it form a continuous layer over the surface, so the mosquito, they do not get the water surface for oviposition or laying egg. So that is why it is used as a control measure for preventing mosquito breeding. So, it has got another very important ecological role, that is bioremediation. Azolla pinnata and many other species of Azolla as well, they have got a very high tolerance for heavy metals as well as organic dyes such as methyl violet, rhodamine. So these are present in the wastewater, and they should be removed, because some of them are that, especially the heavy metals. Heavy metals are accumulative poison, so they create metabolic disorder in human as well as other organism. And these organic dyes, some of them are quite carcinogenic. That is why Azolla is used very successfully as a bioremediation plant. Azolla, it's a very fast grower, so it is a very good source of for reducing the carbon dioxide in the air. It is a very good source of carbon sink, much good source than the mangroves also. Mangroves are also a very good carbon sink, and Azolla or any other fast growing fern is also. So the this very fast growing biomass, it can be used, it can be harvested and used as to produce biofuel. And that mass of Azolla can also be used for nitrogen fixation, especially in the rice fields. So this is how um, this azolla cultivated in India for various purpose, like for biofertilizer mainly, and also for biofuel, as well as food and
Then
fodder
we see
for the
that
animals. This azolla has got a very important secondary metabolite, that is deoxyxanthocyanin. This is actually secondary metabolite that is produced in large amount, triggered by herbivory. So once there is herbivory, amount of this secondary metabolite, it increases, and this is just a reaction to prevent herbivory. Then about the control measure, this weevil Stenopelmus rufinesus, this weevil is a biological control measure for azolla. We know that azolla, it accumulates a large amount of heavy metals and organic as well as many inorganic pollutant in the wastewater treatment. So it is very much resistant to different sort of chemicals, and so it is a better way to biologically control azolla by the use of this weevil. And azolla, most importantly, it is used as a model organism to study their symbiosis. Many Cretaceous fossils has been found of azolla, and one of the most interesting feature is that in even this fossil azolla, anapina has been found. So this anapina and azolla inter symbiotic relationship has been even in the Cretaceous period also, where we find most of the fossils, and this fossils has been found throughout the world. In India, we find the fossil of Azolla intertrapia, and in America, that is Wyoming, we find fossil of Azolla berrii, and in Europe, that is England, we find the fossil of Azolla presca, and again in Nevada we find fossil of Azolla tertia. We also find it in the in Canadian region, that is in the here also we find Azolla primevia, and also we find um, in the South America, that is in the Bolivian region, we find Azolla boliviensis. So
most
basically
of
we
the
see that
fossils they
most of the
belong
fossils are
they
found in the
belong to
American,
the Ecocene.
European and Asian
uh,
continents.
Here
That is this region, in during this epoch, most of the fossils are found. However, some fossils are also found in the Oligocene, that is here, as well as in the Paleocene also. So we see that basically these three epochs we find the Azolla fossils mostly. So this was the first part of the video where I discussed the distribution, the morphology, anatomy, various ecological and economic importance of Azolla and the fossil record. Next, I will be discussing in the second part of the video, I will be discussing reproduction and life cycle of Azolla. I have also made videos about other type of water ferns and also about the land ferns, and description of in the description box I will be putting all the links of those pteridophyte videos. Also, I have made videos about algae, about fungi, lichens, cyanobacteria, bryophytes, And I have also started making videos on on ecology and the MSc and BSc syllabus. And please do write in the comment section if you want me to make any new videos. And those who have already made commented on the videos they want, I'll be making all their videos one by one. And thanks again for commenting and subscribing. And those who are new to my channel, please do subscribe. If you like my video, please do share it with your friends so that it can get more views. And thank you again for watching this videos. So next video will be on the second part of this Azolla.